To the structure of the male gametophyte, now we move on to the structure of the female gametophyte. Now before I begin with that, first and foremost let us quickly revise the structure of the female part of the flower, that is the carpel or the pistil. So each carpel or pistil is made up of a flat expanded portion called the stigma. This is the landing place for the pollen grains. The pollen grains land here at the time of pollination. Then we have the slender style that connects the stigma to the ovary and a swollen basal ovary. This ovary is divided into chambers, and these chambers are called locules, and the locules contain, suppose these are two chambers in the ovary, the locules contain ovules. Now, development of an ovule. This ovule arises as a knob-like structure in the ovary, in the wall of the ovary, and as it develops, it rises up to form a stalk-like structure which is called the funicle. So, if you look at the structure of the ovary, suppose this is the ovary. From the wall of the ovary, the development of the ovule arises, and the stalk-like structure on which the ovule is based is called the funicle. Now, this funicle is going to hold the ovule, and this point of attachment of the ovule to the wall of the ovary, this point of attachment is called the hilum. This is the funicle, stalk-like structure on which develops the ovule, and this is the point of attachment. This multi-layered tissue is the nucellus. This is then covered with two layers of integuments. These are two integuments. Outer, this is the outer integument, this is the inner integument. And these integuments do not cover the entire nucellus. They are open at a point, and this point is called the clasa. This ovule contains the embryo sac, and the embryo sac is the female gametophyte. We are going to see how the embryo sac develops, what is the structure of the embryo sac and how its development takes place. This is the embryo sac, and it's the embryo sac which contains the female gamete called the egg cell. In the embryo sac, this is the egg cell or the female gamete. And actually, the embryo sac is monosporic in nature. It is an 8-nucleate structure, and these 8 nuclei are arranged in a very definite manner. This opening in the integuments here is called the micropyle, and towards the micropylar end there are 3 nuclei. The central is the egg nucleus, and all these 3 nuclei together form the egg apparatus. Egg nucleus, or we also call it the egg cell. These two surrounding the egg cell are called synergids, and 3 cells are at the opposite end towards the clasa. 3 cells are here, 3 cells are at the micropylar end. These 3 cells at the clasal end are called the antipodal cells. 2 nuclei are present in the center. These are the 2 polar nuclei which then fuse. These 2 polar nuclei fuse to form the secondary nucleus. So actually there are 8 cells in the embryo sac which finally become 7 cells. So we can say embryo is monosporic, 8 nucleate structure which changes into 7 cells because these 2 polar nuclei fuse to form one secondary nucleus. So this is the structure. And how does this develop? So one cell in the nucleus, one cell in the nucellus, this multi-layered tissue around the embryo sac, one cell in this differentiates and is called the archisporial cell. This cell is actually the megaspore mother cell, because the megaspores are going to arise from this archisporial cell by meiosis. This archisporial cell is diploid, that is the megaspore mother cell is diploid. It undergoes meiosis and it forms 4 megaspores. All this development is taking place inside the ovule, and ovule is the megasporangium. Ovule is called the megasporangium, and the development of the megaspores are taking place in the megasporangium, and this process of development of megaspores in the megasporangium is called megasporogenesis. Genesis means formation. Formation of megaspore in the microsporangium is called megasporogenesis. So 4 megaspores are formed. Out of these 4, 3 will degenerate. 3 will degenerate and only 1 will be functional. And this functional megaspore will finally form the embryo sac. It will divide mitotically and will form an 8-celled structure. This 8-celled structure is called the embryo sac. The 8 cells organize themselves in a definite manner inside the embryo sac. The embryo sac is called the female gametophyte, which will contain the female gamete. So these 8 cells which have been produced mitotically from the functional megaspore will reorganize themselves in such a manner that 3 are towards the micropylar end and 3 are at the classal end. The ones that are near the micropylar end, 1 is called the egg cell or the female gamete, which are surrounded by 2 synergids, and the 3 at the classal end are called antipodal cells, and the remaining 2 are called polar nuclei, which are in the center of the embryo sac and which fuse to form the secondary nucleus. So this is the structure of the embryo sac, which is monosporic 8 nucleate structure. Later changes to a 7-celled structure. So this is about how the ovule develops inside the ovary and how the female gametophyte, that is the embryo sac, develops inside the ovule, which is also called the megasporangium. The process of development of embryo sac or the female gametophyte from the megaspore, mother cell or archisporial cell is called megasporogenesis. So this was how the female gametophyte develops. Now I will also tell you one more important thing, that the ovule that is formed, the structure of the ovule we have discussed here. This ovule that is formed can be of various kinds depending upon the position of the micropyle and the classa. The arrangement of the micropyle and the classa, there can be different kinds of ovules. Now these ovules, first is orthotropous ovule. Ortho means straight. The word ortho means straight. In this ovule, this is the embryo sac, nucellus, integuments, leaving an opening in the center. So this is the micropyle, this is the claza, and this is the funicle. Micropyle, claza and funicle are all in one straight line. The three are in a straight line. So this kind of ovule is called orthotropous ovule. Second, anatropous ovule. Ana means backward. This kind of an ovule is such that the ovule is bent, and the ovule is bent and the micropyle, this is the micropyle, this is the funicle. Micropyle and funicle are close to each other. They are close to each other, and this is the claza. This is the most common type of ovule in the angiosperms. Most common type of ovule is the anatropous ovule. Then another kind of ovule is called amphitropous ovule. Amphitropous ovule is such that the ovule is placed transversely and is at right angles to the funicle. Suppose this is the funicle, these are the two integuments covering the nucellus, this is the embryo sac, this is claza, this is micropyle, and this is funicle. So the ovule is placed transversely and is at right angles to the funicle. The fourth kind is called campylotropous ovule, where the ovule is twisted and the micropyle, suppose this is the nucellus, this is the nucellus, this is the micropyle, this is slightly twisted like this. So the micropyle and the claza are not in the straight line. This is claza, this is micropyle. Micropyle and claza are not in a straight line, and the funicle and claza are at right angles. The funicle is at right angles to the claza, and micropyle and claza are not in a straight line. Then the ovule is called campylotropous ovule. So we have done the structure and development of the ovule, structure of the female gametophyte, and different kinds of ovules in a flower. This is enough for this class, where we have discussed the structure of the male gametophyte and structure of the female gametophyte. So we will be doing pollination on the next turn. Good luck till then.